Okay, everyone's favorite lab, protein folding. Okay, so I'll jump into the specific types of amino acid side chains that determine how a protein actually folds. But almost all of it has to do with the laws of polarity. And so what polarity is a difference in charge. And so water actually has a difference in charge, whether you're talking about the hydrogens, which are partially positive, or the oxygen, which is partially negative. And so polar molecules are attracted to other polar molecules because you all know negative and positive attract to each other, which I guess means if you're a really negative person, you need to date someone who's positive, and if you're a really positive person, try to find someone who's negative. Just kidding, negative people need to be eliminated. So anyways, what you want to do is, or what happens is, water is very polar, and so the behavior of the protein is determined by the fact that it's surrounded in a polar environment. If the protein was folding in oil in a non-polar environment, the laws would be the exact opposite, okay? So they attract to each other. So let me walk through each of the types of side chains. So we have a polar molecule, which is attracted to the water. We have a non-polar molecule, which is not attracted to the water at all. We have a, blue was basic, I believe, which is positively charged. We have an acidic, which is negatively charged. And then finally we have green, which is the cysteines, which basically only behave with themselves. All right, so let's start with the two that are interacting with water. So the polar molecules, they like water. So it's very simple. As the protein emerges, imagine my hand is the ribosome, the protein emerges one amino acid at a time. And so if the amino acid that emerges is an actual polar amino acid, then what ends up happening is it's immediately surrounded by water and it's super happy, right? It likes water, it's polar, it's surrounded by it, and so it's going to end up staying on the outside of the molecule, right? But if the next one is non-polar, it's instantly like, oh, I totally hate water, get me out of here. And so when you saw the video, it was like shaking like this. It's basically the tension of the non-polar amino acids that are trying to get away from it. And so what they end up doing is they end up hiding in the middle of the protein with each other. So all non-polar amino acids end up hiding in the middle, and then your polar ones end up on the outside. So polar inside, non-polar inside, polar outside. All right, so next, the acidic and the basic have to do with magnets, right? So just like I already said, positives and negatives attract each other. Well, that's why negatives basically attract. They're opposite charges, so they end up sticking. And if you have the same one, they repel each other. So what ends up happening is a basic and an acidic come together, and then the other basic would be repelled by that, and so it would kind of move away, and then this one would come in. And so you would have the two acidics, or you'd have an acidic and a basic, and an acidic and a basic coming together. All right, finally, the last type is the green, which is a cysteine. And so cysteine bonds with cysteine. So at the very tip of one cysteine is a sulfur. At the tip of the next cysteine is another sulfur, and so the two sulfurs come together to form a disulfur bond. And by the way, one of the 6 molecules of life is sulfur, and so be aware that sulfur is primarily in proteins. It's not really in any of the other molecules. So anyways, remember sulfur is in proteins. All right, thank you for your attention and good luck on the AP exam.